Cytoskeleton, which is the network of fibers in the cytoplasm that regulates structure and motility, is made of three parts: microtubules, microfilaments, and intermediate filaments. Microtubules are made up of two polypeptides, alpha-tubulin and beta-tubulin. Microtubules grow in length by tubulin dimers, or the combination of alpha and beta-tubulin, being added to the ends of the microtubules. Microtubules are shaped like tubes, as their name suggests, and look like this. In many cells, the microtubules are centrally located around the centrosome, which acts as an organizing center for microtubules. Microtubules near the centrosome come together and form centrioles. Microtubules have one main function, which is to shape and support the cell internally. They are the equivalent of steel girders in a skyscraper. In eukaryotes, microtubules are responsible for the beating of flagella and cilia. Without the tubules, we would not be able to move debris from our lungs, nor would our blood cells be able to exit the blood vessels.
Okay, microfilaments, otherwise known as actin filaments, are thin thread-like protein fibers about 6 nanometers in diameter, and they are made out of a contractile protein named actin. And actin and myosin work together in muscle contractions. Microfilaments assist in cell division and also anchoring down proteins inside on the plasma membrane. They also assist in maintaining cell shape and movement. They are usually found on the outside edges of the cell, and without them, the cell would lose its mobility and its shape. They're also important in the structure of pseudopodia, and they are very important, like I said, in cell division and streaming of the cytoplasm, which is when there's a current in the cytoplasm that organelles and materials can move around in. One interesting fact is that they grow faster on one side than the other, and like microtubules, they also are able to construct and deconstruct very easily. And these are some models of microfilaments. They're like long, thin, and very sturdy, as compared to an intermediate filament, which is obviously thicker. Alright, intermediate filaments are fibrous proteins that are coiled into cables that are about 8 to 12 nanometers thick. They're made of keratin proteins, and they're in pairs, one's acidic and one's basic. Their function is to maintain a cellular shape, and specifically to maintain the tensile strength of the cell. They anchor down the nucleus and other large organelles like the mitochondria as well. And because of that, they're usually found toward the middle of the cell. Mutations, an interesting fact is that mutations in intermediate filaments can result in neurological disorders as well as blistering diseases. The structure of the intermediate filament is much more permanent than either microtubules or microfilaments, and they don't deconstruct as much or as often as microtubules or microfilaments do. And without them, the cell would lose its internal organization. And just as a comparison, this would be microfilaments right here. This would be a microtubule, and these would be the intermediate filaments, because they're intermediate between the microtubule and the microfilament.